Fission yeast may not have arms, legs, or existential dread, but in the world of aging research, it punches far above its microscopic weight. These tiny single-celled organisms, specifically Schizosaccharomyces pum, have been affectionately nicknamed mini-humans in the lab. And while they don't sip lattes or forget where they left their keys, they share a surprising amount of genetic and cellular machinery with us, especially when it comes to aging. So why study a humble yeast cell to understand human longevity? The answer lies in simplicity and conservation. Fission yeast has a streamlined genome and fast life cycle, but it retains many of the same core pathways that regulate aging in higher organisms, especially nutrient sensing, DNA repair, stress response, and cell cycle control. Think of it as a stripped-down version of the human cell, without all the complicated noise. That makes it easier to tease apart the specific genes and molecular levers that control aging. And unlike studying people, you can do a full lifespan experiment in yeast over a weekend and not get sued for unethical experimentation. One of the most groundbreaking things fission yeast has helped reveal is the role of nutrient-sensing pathways, particularly the TOR, target of rapamycin, pathway, in regulating lifespan. In yeast, dialing down TOR activity through genetic tweaks or nutrient restriction dramatically extends life. Sound familiar? That same TOR pathway also exists in humans and is now a central focus of aging research, with potential implications for treating everything from cancer to neurodegeneration. But here's where it gets even more interesting: caffeine. That ubiquitous molecule fueling your daily productivity is also doing some sneaky things to yeast, and possibly your cells too. Studies in fission yeast have shown that caffeine can extend lifespan, largely by inhibiting the TOR pathway and mimicking the effects of caloric restriction. Basically, caffeine fools the cell into thinking it's living in a nutrient-scarce environment, even if it's swimming in sugar. This triggers a low-energy, stress-resilient state that favors repair over growth, a hallmark of extended health span. Genetic studies in fission yeast have also helped identify which genes interact with TOR and AMPK, the pro-longevity, energy-conserving enzyme, to regulate lifespan. This has opened the door to a deeper understanding of how genetics influence aging, and how we might manipulate those levers with drugs, diet, or lifestyle changes. Some of these genes have homologs in humans, meaning the insights gained from yeast aren't just theoretical, they have real potential for translation to human biology. Fission yeast has also provided clues into how cells manage stress, how they repair DNA, and how epigenetic changes, the kind that influence gene expression without changing the underlying DNA, accumulate over time, all key aspects of aging. And because these cells divide and age similarly to how our own cells do, scientists can study things like senescence, biological aging at the cellular level, and longevity interventions in real time. In essence, these mini-humans are giving us a cheat sheet for our own biology. They've shown us that aging isn't just wear and tear, it's regulated by deeply conserved genetic programs. They've hinted that lifestyle choices, like consuming caffeine or periodically restricting food intake, might influence those programs through pathways like TOR and AMPK. And they're proving that even the smallest organisms can teach us some of the biggest lessons about how to live longer, better, and maybe even a little more like a well-optimized yeast cell, minus the fermentation, of course. So the next time you drink a cup of coffee and skip a meal, know that somewhere, in a glowing petri dish under a microscope, a fission yeast is doing the same thing, living longer and passing its secrets along to science.